Good morning, Dokter Sukanya. Nice to meet you.
Assalamualaikum.
Wow,
Waalaikumsalam.
Waalaikumsalam.
Halo dokter.
Sorry that I could not change the background. Yesterday
Okay, it's
I
okay.
said that I did it already, but now it's not coming.
Yes.
So how to change it? Moment.
Okay.
So few minutes.
Please, Dokter Ika, dia Ika.
Oh, okay.
Yeah, that's nice. Sukanya.
Sorry.
It's okay.
Yeah.
Okay, we will open this case lecture. Amalia Zahra will be the MC. Okay. Okay, Amalia.
Okay. Thank you to Prof. Sukanya and also Prof. Syamsuddin and also Bu Dia Ika and Bu Yuslia for allowing me to open this guest lecture today. Assalamualaikum warahmatullahi wabarakatuh. Shalom, Om Swastiastu, Namo Buddhaya, Salam Kebajikan, Salam Pancasila. Good morning and warm welcome from Indonesia, the Honorable Dean of the Fakulty of Farmasi Universitas of Pancasila and FIC, and also the honorable speakers and moderator of international guest lecture today, and all participants who joining us online in this guest lecture today. Before we begin the opening ceremony, allow me to introduce myself. My name is Amalia Zahra Apriliana, as the Master of Ceremony today. This is a grateful opportunity for all of us. As we know, around a month or 2 months ago, we just had the first series of international guest lecture with topic pharmacology and clinical pharmacy. And the second topic is social and administrative pharmacy. And luckily, now we have another series of international guest lecture with topic pharmaceutical, biology, and pharmacognosy. And of course, we are so excited to hear our speakers to discuss with us today. And ladies and gentlemen, before we continue to the next agenda, we would like to invite you to sing the national anthem of Indonesia, Indonesia Raya, followed by hymn of Universitas Pancasila. For all participants, please be seated properly.
Universitas Pancasila, tetapkan hati masa depanmu secemerlang mentari. Tetapkan hati para dosenmu berjuang untuk keberhasilanmu. Dari kampusmu kau kan sebarkan ilmu, dari kampus pula kau himpun ilmu. Kau tegakkan, kau siapkan, kau hadapi masa depan. Bawalah pandangan dari rakyatmu. Di sana untuknya dituntut namamu. Di sana untuknya dituntut baktimu. Jangan kau silau pada dunia ini. Langkahkan maju, tundukkan rintanganmu. Hanyalah satu tugas muliamu. Manfaatkanlah diri untuk sesamamu. Dari sini ciptakan dunia aman damai. Dari sini ciptakan dunia aman damai.
Thank you. Up next, we would like to invite the Dean of Faculty of Pharmacy of Universitas Pancasila to give a welcoming speech and opening remarks. To the Honorable Professor Dr. Apoteker Syamsuddin Magister Biomedik, the time is yours.
Thank you, Amalia Zahra. Good morning, everyone. Assalamualaikum warahmatullahi wabarakatuh. The Honorable Associate Professor Dr. Sukanya Di Ajisai and Professor Dr. Mogana Sundari Raja Gopal as speaker, the Honorable Lecturer, students, and participants. As the Dean of Faculty of Farmasi Universitas Pancasila, I highly appreciate the presence of Associate Professor Dr. Sukanya from the Department of Pharmacognosy and Botany, Faculty of Pharmaceutical Science, Prince of Songkla University, Thailand, and Professor Dr. Mogana Sundari Raja Gopal as the Dean of Faculty of Pharmacy, UCSI, Malaysia. I got to know Associate Professor Dr. Sukanya during training grants on the specification and standardization of medicinal plants and related products on 19 June 2015 at Prince Songkla University. Prof. Sukanya is a friendly and the expert of the real and natural product. And topic presented today is the herbal research for product oriented. Maybe in the future we can collaboration, Dr. Sukanya.
Thank you. Yeah,
Nice to meet
welcome.
you.
Yeah.
Professor Dr. Mogana Sundari Raja Gopal is also an expert in this field, whose field is the same as mine in the development of natural medicine. Topic presented today is the application of pharmacognosy in plant-based antimicrobial and anti-tumor. I seriously hope the Faculty of Farmasi Universitas Pancasila can cooperate with UCSI for the doctoral double degree program. This international guest lecture activity is one of a series regarding Faculty of Farmasi Universitas Pancasila 61st anniversary. Therefore, I truly optimize this guest lecture to provide new perspectives, inspiration, and the deep understanding for all of us. I encourage the participants to actively participate as and have discussion during the lecture. Let us welcome this guest lecture with great enthusiasm. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Professor Dr. Apoteker Syamsuddin Magister Biomedic, for the wonderful speech. And up next, we continue to the next session. It's a photo session. So for all participants, please turn on the camera. Okay, to capture this moment is gonna lead by me. So for all participants, please be ready. Put your best smile on this moment. Okay, I'll capture it in 1, 2, 3. To the next slide. 1, 2, 3. Okay. Okay, it's already captured one by one for this slide. Thank you so much. And then now we have moved on to the main event of our meeting today. As Professor Syamsuddin just talked about, that pharmaceutical biology and pharmacognosy are two closely related fields that delve into discovery and analysis and application of natural products in the development of drug and therapeutics. And these fields face several challenge for us, and including the sustainable sourcing of natural materials, and then the need for more analytical techniques that we need to know, and then overcoming the complexity of natural product structures. And these natural products can contribute to modern medicine, are invaluable, offering hope for treating various diseases and conditions. And what we can do as a pharmacist to contribute to this field, it will be answered today. But before we begin our lecture, I would like to read the curriculum vitae of our moderator for today, Apoteker Michael, Magister Farmasi. Apoteker Michael, Magister Farmasi, is an analytical method development and preclinical manager at Soho Global Health Jakarta, and he graduated from Universitas Pancasila for bachelor, pharmacies, and also for master program. He has work experience around 9 years as analytical method developer, and he also focused on herbal medicine and supplement product. So without any further ado, now already joining us with us. Good morning, Mr. Michael.
Hello, good morning, Amalia, ladies and gentlemen, everyone.
Okay, by that, the floor is yours.
Okay, thank you, Amalia. So, good morning, ladies and gentlemen. My name is Michael. It's been an honor for me to be your moderator today for today's webinar about pharmaceutical biology and pharmacognosy. First of all, I will briefly introduce our speakers for today's webinar. Our first speaker is Associate Professor. Sorry. Okay, I will continue. So our first speaker is Associate Professor Sukanya from Prince of Songkla University, Thailand. So Prof. Sukanya obtained her Bachelor of Pharmacy in the Prince of Songkla University, and then she graduated master degree from Chulalongkorn University, majoring pharmacognosy, and her doctorate degree from Robert Karls Universität Heidelberg, Germany, majoring pharmaceutical biology. Currently, her position is a lecturer and pharmacognosy, pharmaceutical biology department head from 2007 in the Prince of Songkla University. Prof. Sukanya already published more than 22 papers, Scopus publication, and from 1997, and her latest work about, if I read about cannabis sativa, the weed, right? So today, Prof. Sukanya will present about the herbal research for product-oriented. Before we begin, I will read some rules. If the participant wants to ask a question, you can type the question in the chat box in English, and I will gladly read your question to Prof. Sukanya. Prof. Sukanya, you will have 30 minutes to deliver your presentation, and after that, we will have 10 minutes of Q&A session. So without further ado, ladies and gentlemen, please welcome Prof. Sukanya. Prof. Sukanya, the time is yours.
Yeah, thanks a lot, Michael. Could I share my screen by myself?
Yeah, Prof.
Okay. Could you see my slide?
Yeah, clearly.
Okay. Okay. Thanks a lot for University of Pancasila that invite me to be the guest speaker today. I have 30 minutes, you said to me. Okay, so should we have to start? After I have got the invitation, I think that what is a topic that is suitable for the student and also for the lecturer or researcher in Pancasila. So I think the important now is the herbal research for product oriented. Because in Thailand, we have the question that now we just have only the research only. But after that, it stopped on, we call that stop at the table on the shelf. Is not proceed to the company or to the products. So now that's why we have to think about after the research, how they can go on. So why that is come this topic. So for the content of this, my speech is we will include this topic is introduction about herbal research and the example. The example for the research, I will show you about the enzymatic assay because I think this topic will be easy to understand and is easy to do in the lab and to proceed until you get the products. And I will show the example for the research about enzymatic assay. The first one is a herbal research for whitening. Whitening is mean, I think this one is will be interesting for the student. Normally, it also in Thailand is also interesting for the student for to be a cosmeceuticals. And the second one is will be for the disease is anti-diabetics product oriented. And after that, I will show you the example of the herbal products based on the research. The first one I will give you to the idea for the research of natural products. Normally we start by screening. How to screening is mean that in Thailand, I think also in Indonesia, we also have a lot of herbal medicine, and how we choose that. Which one that we will choose to be for the research. So we have to start with the screening. And after that, we will do the bioassay or isolated the pure compound and continue once more for the bioassay. And after that, we will choose the best one to continue with the in vitro or to do the prototype formulation or clinical research. And sometimes now I think the good way also that you have to check the correlation between the compound or the bioassay with molecular docking. What is molecular docking? It means that you use a software for the computer in the computer, and after that you take the receptor and also the chemical and fitting about that and you can look the relative of the activity together with the bioassay. So this you will get the the potential of the chemical compound from natural product to be the medicine in the future. From the research to the medicine or to the product, we will start with the in vitro model. I think you know about in vitro. In vitro is mean that you don't work with the animal or with the human. You work only in the tube in the lab. And for the in vitro model, we will do for the enzyme-based assay. and also the cell-based assay. What does it mean? It means that this you work only in the lab, and after that, after you get the potential one, you can continue to the in vivo model. And after that, you can continue with clinical trial. This is the idea for the medicinal plant research. And I would say that you have to start also for the phytochemistry. Do you know for this? I think for pharmacy student, maybe you learn a lot about phytochemistry from natural products. It means that you look for the chemical constituent in the plant. How to look that? You look about the chemical and physical property of the plant chemical compound. And also you can continue with extraction, isolation, and purification. And after that, you also analysis and identify the chemical compound after you isolate it. And somebody, they also study about biosynthesis pathway. This one is a scope of phytochemistry. And for the phytochemistry, normally you can work based on two ways. The first way, you can work based on pharmacological activity. This one is mean that you can screening of biological determination. For example, if you want to look about the plant that has the whitening effect, you can test by do anti-tyrosinase activity. If you want to look about anti-diabetics, you also can test with anti-alpha glucosidase activity. To these activities, I will show you later. And for after screening the biological assay, you can select the plant from the potential one, or sometimes you can select from the folk medicine, from traditional medicine, to continue with the extraction and isolation the active compound. And the second way that you can work based on chemical constituent. The first one, you work with the pharmacological activity. The second one, you can work with the chemical constituent, because sometimes the research group, they're looking about only the compound in each group. For example, some group, they say that, oh, I interest only alkaloid. The other group, they say that, oh, I interest only flavonoid. So they try to screening and look only, focusing only one group. And this why you can select on only one group, because we have the assay to check the group of the chemical compounds. After you select that, the isolated active compound, and after that you can take this isolated compound to do the biological activity later. And the important for phytochemical screening, the first one is must be fast, because you want only screening to select the best one. So the first one is have to be fast, and second one is must be easy and need less of equipment, and specific only the wanted chemical group. If it's specific, it will be good. You can look at the chemical one. And also that it would be good if it can able to approximate the amount of chemical substance. And also the best one, if it can identify the chemical compound. But the number 5 and number 6 is, I think it's quite difficult for phytochemical screening, because just only for screening. But I think the best one is must be fast and easy. That is important. And the benefit of phytochemical screening. The first one, we can know the chemical groups that are present in the plant. For example, that if you screening for alkaloid, and after that they show the positive for the alkaloid reagent. So after that you say, oh, this plant has alkaloid. You can select it and continue your research. And also that for the phytochemical screening, you can predict the pharmacological activity of this plant. For example, if you screening and you found that, oh, this plant has a flavonoid, you can predict that it can have the antioxidant activity. It can be. Normally flavonoid, most of them has an antioxidant oxidation activity. So that is you can predict that. And also that you can know how to extract the substance from the plant, because when you know the group of the chemical compound, you can know how to extract, because they has many methods for extract the plant. And after that, you also can select the plant for further phytochemical study for the interesting group of your sample. And the best one for the advisor or supervisor is normally phytochemical screening is more cost effective. This is important because if you work with biological screening, it's more expensive than phytochemical screening. This is important because advisor or supervisor have to pay for this one. Yeah. And I will show you the second one is this is a biological screening. It's a one-off biological screening. Is an enzymatic assay. That is most of my work at the moment in the lab is about enzymatic assay. This is a laboratory method for measuring enzymatic activity. As you know that you can detect also enzyme kinetic and enzyme inhibition. And normally they has many types of assay. When you want to determine, it depends on you that you want to determine the consumption of the substrate or the production of the products. Yeah, you can do this one or the other one. Yeah, and normally for enzymatic assay, they have different methods to measuring the concentration of the substrate and product. Yeah, and it's easy to do this method. And they divide into groups. It depends on the sampling method. If you want to continuous assay, it means that you can continuous leading the activity. So it means that you start the experiment and you have to run the method continuous, and you record the data on times. But the second way is this continuous assay is opposite. Opposite is mean that you have to read the result from when the reaction stop. For example, you say that you will run this method for 20 minutes. It means that you will read the result from the assay at 20 minutes later. But for continuous, you will read every second or every 20 seconds until 20 minutes. This is different way. And for the for the first one is a continuous assay. This one is most convenient because it's a one way giving the rate of reaction. That I say to you that no need to stop. You can record the machine, the instrument will record the result on the time that you setting the program. Yeah, and you can also record the substrate or the product during the assay. And the second one is discontinuous. It means that samples are taken from an enzyme reaction in the interval. Interval is mean that you can say that You want to record the data at 10 minutes or 20 minutes or 30 minutes, up to you, that you want to record the result from that. And most of the enzymatic assay we will do with the spectrophotometric assay. This is an easy way in the lab. I think also in Pancasila also have this experiment to do with a spectrophotometric assay, because we can use a UV. This is we look how much the light that the assay solution absorb. You can see this is a basic instrument. They will have the light source, and after that they have monochromator and select the wavelength that you want to know that the sample can absorb that. And after that, you can record the data for the OD. Normally, the reaction for the sample, we have most of them we like to have the assay that has a colorimetric assay. It means that they has the color for the product or for the substrate anyway. But if they has the color and you can see by your eyes, this is good, because after the reaction you can predict when you see by your eye that is active or not active sample. Yeah, but anyway, that is only beginning of your determination by your eyes. But anyway, you can use the UV record the result. That is easy to record and compare the result of each sample. Anyway, they has the other assay also, that volumetric assay. Also, you look the fluorescent and also chemiluminescent assay. They also have, at this you need the special instrument. And I would say that they has many factors that interfere the enzymatic assay that you have to be careful. The first one, the salt concentration. Why I have to say that the salt concentration is important, because most of the enzymes cannot tolerate the extremely high salt concentration, and the ion interfere with the weak ionic bond of the protein. As you know that in the enzyme is a protein, so it's also effect for the enzyme. Typical enzymes are active in the salt concentration of 1 to 500 millimolar, not more than this, except exceptions such as halophilic algae and bacteria. This is the lightest high salt concentration, only this one. But most of them is cannot tolerate with the salt concentration. The second factor is the temperature. On enzyme work with the range of temperature specific to the organism. Increase the temperature is generally increase the reaction rate. It means that if you heat in the experiment, if you set the program to increase the temperature, normally the enzyme is more active. But they has the limitation. They has the limitation of the temperature. It means that if the temperature is too high for them, they can decrease the rate of reaction also. Yeah. Why? Because the high temperature can denature the protein structure to break down the weak ionic and hydrogen bonding of the enzyme. So the enzymes you can see from this picture, the active function of protein, they have the picture like this. But if you heat it, they become the denatured protein like this, that is cannot function. And normally the optimum temperature for human enzymes is usually about 35 to 40 Celsius. As you know that our body temperature also average about 37 degrees Celsius. Yeah, and so human enzyme, when you work with the enzyme, you have to be careful. If the temperature above 40 degrees Celsius, this can be the denature of the enzyme protein. The third one is the effect of pH. Most enzymes are sensitive to pH and have specific length of the activity, so they have the optimum pH. Before you start the experiment, you have to look the suitable pH for each enzyme that you want to work. Because the pH can stop the enzyme activity, also they can denature the enzyme by breaking ionic or hydrogen bond. Most enzyme function is between pH 6 to 8. Yeah. However, they have some special enzyme. For example, pepsin. Pepsin, as you know, that in stomach. And this is the base pH for working of this pepsin is a pH 2, is acid pH. But trypsin, as you know, that in the intestine, this is working about pH 8, is a basic pH. And the other one is substrate saturation. Increase the substrate concentration, increase the rate of reaction also. However, the enzyme saturation limit the reaction rate. As you know that the enzyme is saturated when the active site of the molecule are occupied. You can see this model, this is enzyme, this is substrate. Normally when they're working, they have to fitting at the enzyme receptor. And if the work is mean that on the receptor active site of the enzyme is fitting already, so that is the limit of working of the enzyme. So the saturated point, the reaction will not speed up. Normally, when you increase the substrate concentration, enzyme activity also increase. But if the receptor of the enzyme is occupied already, so if you add more substrate, nothing happen, nothing increase. The enzyme activity normally that we know that we call the compound that influence the rate of the enzyme catalytic reaction are called modulator or modifier. Usually the effect is to reduce the rate of the enzyme. We call it an inhibition. And sometimes the enzyme reaction also increase the substance that can increase the enzyme. We call also activator. But most of the compound from natural product that we want to make the research study is we looking for the inhibitor from natural product. Generally, classification of enzyme inhibitor. As you know from this figure, you can see that the enzyme inhibitors are divided into two ways. First one is specific. The second one is non-specific. For the non-specific, it means that on of the enzymes can stop working by this factor. For example, if you add the acid, strong base, or you add ethanol, heavy metal, reducing agents to your experiment, the enzyme on-off the enzyme can stop working because it's non-specific for each enzyme. But for the specific, it means that some enzyme can stop working, the other maybe not stop because it's specific for each enzyme. For specific, we divide into one is reversible, the other one is irreversible. For irreversible is we divide into is one is suicide inhibitor. Suicide inhibitor is mean that when you add this compound to the experiment that has an enzyme, they can stop on the enzyme and cannot reverse back to working anyway. It's dead, completely dead. And the second one is time dependent inhibitor. It means that it's a take time. It's take time to stop the reaction of the enzymes. And most of the study we will do the reversible. Reversible is we divide to look the mode of action. The first one is competition. The second one is non-competition. The third one is uncompetition. So if you want to know about this, you have to make the experiment to look that your compounds, how it's work to the enzyme. And so most of the study for enzyme, we will continue to look about this. And have the warning when you work with the enzymes, you have to be careful about tannin. Tannin, normally you know that's on the medicinal plants has the compound because tannins is how to say, normally for the leaves, for the bark, for the stem, they has a tannin. And when you have the plant extract and you taste with the enzyme, tannin can precipitate the protein and the enzyme also the protein. So it's like that the tannin precipitate the enzyme. So you can get the result that is false positive. So it means the group that can work is a tannin, not the other compound in the plant. The second one that you have to be careful is the color. The color of sample crude extract. Sometimes when they have a strong color, it can be overflow for the microplate reader, the UV instrument, so you cannot determine the result. And the concentration at your screening, and when you working, if you work with a small amount of the compound, you have to use a micropipette. And you have to be careful if you have no experience about micropipette. Your standard deviation will be wide. Normally, you have the average, the result, and you have the SD, standard deviation. So if your data is not good, the standard deviation will be wide. And the timing you have to choose, continuous or discontinuous assay. And when you work, you have to be careful also that your sample have to be the homogeneous of the sample code extract. Sometimes my students take only the supernatant and the active compound is precipitate at the bottom of the cuvette, something like that. So it means that you take only supernatant is not working, it's not active because the active one is precipitate, done homogeneous sample. And also the concentration of the solvent. Most of the solvent that we use in the lab is the DMSO. For DMSO, you have to be careful because if you use a high concentration of DMSO, it also can precipitate the enzyme. They can denature the enzyme also. So we have the limitation how much that you can use DMSO for each enzyme assay. And don't forget to have a positive control in your experiment. Sometimes the students say, oh, I have positive control already last week. So this week, I don't make the positive control once more. I say, no, you cannot do like that. You must have the positive control every time that you're working. Why? Because if you don't have the positive control, you never know that today that you work, your experiment is okay or not. Sometimes they say that, oh, my sample doesn't work. No samples is working, is active, no one. And if you have the positive control, you also know that today your experiment is failed because the positive control also not active. Yeah, that is why important to control your experiment. You must have this every time that you work. And the cut off, what does it mean? It means that when you screening your extract, you have to make the cutoff. For example, you say that if they have the 80% of inhibition, it means that is active. Or if they have 50% inhibition, it mean active. This is mean cut off of your experiment. How to choose this? Normally, if you work with the low concentration of the sample, for example, if you say that you will screening of biological activity of your sample at 20 microgram per milliliters, and the second experiment, you have the concentration of your sample at 2 milligram per milliliters. So you must have different cutoff because 20 microgram per ml is very low concentration. So you take only 50% is already active, and 2 milligram per ml. So I think that it must be 80% inhibition. That is mean active. That is why it's totally different for cutoff. So now we
Sukanya,
go to the process. Yeah.
Prof, I'm sorry to interrupt you. You only have 5 minutes left on your presentation.
5 minutes?
Yeah.
Okay, so the process of biological activity, this is a flow chart of, you can select the screening of the plant, and this is the guideline, the scope of your work. You can work with the phytochemical investigation, and you can work in the cell line, and after that, you can go to the select one and to do the prototype and clinical trial. And this is the example of anti-tyrosinase activity that I said to you. Because in Asia people, normally we have a dark skin, but we want to be white skin. So normally you melanin pigment is good. It's good to protect the UV, but the problem that when you have the hyperpigmentation, it can make your skin is more dark. For example, melasma or abnormal pigment. That is you don't like it, and also sometimes it's could go to the also the plants, the fruit, something like this. In Thailand, we have a problem for the mercury or hydroquinone that they add in the sample. That is not good in the product, and this can make the problem from your skin if you use hydroquinone and mercury. And so we're looking for the plant extract that can inhibit the tyrosinase enzyme. If you can inhibit this enzyme, you can lower the melanin pigment production. This is the experiment you can see from the flow chart. We can start with the plant extraction, and after that, we look the plant extraction that can stop the enzyme. We call this method is Dukung method. Yeah, and you can look in this publication if you want to read more. And the second one that I would like to show is diabetics. As you know, this symptom already that is so complicated and is now is more and more problem in Thailand. And you can start to work with anti-diabetics activity by in vitro model, and that you can continue with enzyme and also the cell-based assay. And we're looking for the alpha-glucosidase, because if you can stop alpha-glucosidase, you can lowering the blood sugar. And we start the same, that sample preparation, and after that you can macellation, you can extract, and continue with the experiment. The experiment that we have the substrate, we have the enzyme, and we looking for the sample or positive control that is can inhibit this enzyme, and the production of the para-nitrophenols will lowering. The color is will be pale yellow, and this experiment, so I have to go faster. This is microplate liter, and this is 96 well plate that we use in this experiment. And this is when you want to analyze, you can analyze the percent inhibition, and after that you can continue with the 50% inhibition, the concentration, and you can look further in this journal. Yeah, and now go to the last part of my talk is from the research to market. After you select the potential sample, you can go to the prototype formulation and continue with the clinical trial. And this for the example for the product. The first one is a facial mark from the kojic list fungal extract, because kojic acid, we know the best one also for whitening. So we want to make the cosmeceutical for the facial mark, as you see this. And this is for anti-acne facial sleeping mask for Garcinia mangostana. Yeah, we make the experiment and we control with antimicrobial of the product also. And the second one, we make a whitening also, but we make the powder from Dioscorea hispida. Yeah, we make the powder from this, and after that, we make like a face baby powder. Yeah, and we add the extract that has whitening. And this one is henna hair dye cream. We want to use ready to use of the henna dye for the hair. And this also come to the medicine for some problem that they has a problem at the feet, and we combine two plant extract, Cinchibus casumuna and Capsicum frutescens together in this to make like a gel for external use. This is good one, and this we try to make the research for the Korean ginseng Thai herbal mixture tea for anti-diabetics. And this we make like a lozenge formulation for Acmella oligacea extract. Yeah, and so I think time is limiting, so have to say that Limokase, thank you and ขอบคุณค่ะ. And this one, I would like to show that one PhD student from also Indonesia. Now he is a lecturer in Ahmad Dahlan University in Indonesia also. So nice to come to talk today. Thanks a lot.
Thank you, Prof. Sukanya, for the very interesting presentation.
Yeah,
Once
very
again, I'm
sweet.
sorry, the time is very, very limited, so we have to reduce the presentation. So, Prof. Sukanya, if I may summarize your presentation today is about in vitro test to be initial research for a herbal medicine, but the in vitro test should be taken with some precaution, like you mentioned before, the effect of temperature, time, and so on. Yeah, I think your presentation is very interesting. I see many questions already in the chat box. I will jump to the first question, Prof. Sukanya. So the question is from Ms. Farida. The question is, I'd like to ask how to avoid false positive result in extract sample. Thank you.
Excuse me, what do you ask me? Once more, please, pardon.
How to avoid false positive result in extract sample.
You mean that how to find the active one?
I think, Ms. Farida, if you want to explain in detail, you can unmute. I think I will jump to next question first, right? So, Prof. Sukanya, I will jump to next question, to the second question. Question is from Adi Dwi Margono. The question is, In enzyme test using crude plant extract sample, there are several factors that can affect the test result. Explain in detail how factors such as tannins, sample color, sample concentration, incubation time, sample homogeneity, solvent, and positive control can affect enzyme test result.
Yeah.
Good.
Thanks for the questions. Many effects for the result when you're working with the enzymes. For example, that I say to you that when you extract and if your compound has a tannin, normally is the result show is very active because the tannin can precipitate the protein and the enzymes also protein, so they can denature, and so they show the result that's really active. And also that for the how to say the solvent, because when you want to make the experiment, you have to dissolve the extract, and normally you can use the MSO. The MSO we call is a universal solvent. It means that they can dissolve on the sample on the extract, but the MSO, if you use more than 5%, at the final concentration more than 5%, it can precipitate the enzyme also. So it can show you that we call is false positive. False positive is mean that it show that it's active, but the active is active from the solvent, not from the extract. That is we call false positive. Yeah, that is mean that the plant extract is not active. The active is the meso. Yeah, so why we have the limitation of the solvent concentration in the sample. Yeah, and the next one, that is a problem that normally the plant extract, the color is green mixed with the brown with the yellow and green. Yeah, when you dissolve and the color will be interfere the UV absorption of the experiment, and sometimes the instrument, the UV measurement, they show that overflow. Overflow is mean that they cannot detect the result for you. How to solve this problem? You have to dilute the plant extract. Yeah, when you use small amount of the plant extract for your experiment, yeah, the color will be softer, will be pale color, so the work will be better. Yeah, and so that's why when you work with this enzyme assay, you have to be careful many factors, because I work a lot for this, and I see the result and I say, oh, this is a false positive. This is not from the plant extract, something like that also. And sometimes when you dissolve this the sample, the crude extract, the student working, and after that I say that why this sample I think will be active, but it's not active, because I ask the student and I look, the student dissolve is not homogeneous. When they take only supernatant. The extract still precipitate at the bottom of the Eppendorf or the tube. They don't take on of the sample extract to the experiment. Only some part is mean supernatant. That is a problem. I'm not sure that the answer is go to the question or not. Fitting for the question or not.
Okay, thank you, Prof. Sukanya.
Yeah.
To Adi Dwi Margono, I hope that's clarify your question. So the next question is from Andy. What methodologies and techniques are utilized to ensure accurate phytochemical screening, and how do researchers address challenges such as variability in plant material and the presence of interfering compound during screening process?
Yeah, I would suggest that the best one for the solvent extract is 80% of ethanol or alcohol. Normally we use ethanol because if you want to continue with the pharmacology in the animal model, we would suggest to use only ethanol, don't use methanol or the other organic solvent because ethanol is more safe for the animal study, and also for when you want to further with formulation also we would suggest to use ethanol, but the best one you have to use the 80% of ethanol. It means ethanol 80, water 20. Why I would suggest this one? Because you never know that in your plant compounds, chemical compound is a polar or non-polar. You never know that if you don't have the more information that, oh, this plant has only the polar compound, this plant has the non-polar compound. So the best one, you have to use the solvent that can extract most of the chemical compounds from your plant. Is so 80% of alcohol is mean that we hope that it can take polar and non-polar compounds from the plant to in the extract. And after that, after you screening and you looking and you think that this plant is this extract is active one, you can further by working phytochemical investigation. It means you can isolate compound from the plant, and after that, if you get the pure compound, you can come back. You can come back to look the biological activity once more with the plant compound that you can isolate from this plant. And after that, you can say that, oh, this is an active compound. That at the beginning, you screening by use a crude extract. And you don't know at the beginning that which compound in this extract that is active. But after you isolate and you get the pure compound, you have to take this pure compound back to biological determination once more to confirm this compound or not. That is an active one. This is normally we call is phytochemical investigation. We use column chromatography, layer chromatography, HPLC, or the other method in chromatography. Yeah. And also, I think I see the question about cosmetic.
Yeah, Prof. Sukanya.
Yeah.
I think time is very limited. Once again, I'm sorry to all
Okay.
participants.
Yeah.
I think I not be able to read all the question, but last question, Prof. Sukanya, about beauty product. From Rizky Akbar, there is so much item beauty product that we can find in Thailand. What he want to ask you is there any beauty product that made from specific compound or maybe any particular herbal medicine that were regularly used for beauty product in Thailand? Thank you.
Yeah, many products in Thailand that is composed with the medicinal plants. For example, that curcuma longa, turmeric. The yellow color, normally we eat it, we eat it, but now they compose in the cosmeceutical. Yeah, to make for wound healing, for anti-acne, and also mangosteen, Garcinia mangostana. Mangosteen, I think also you have in Indonesia, same as in Thailand. Mangosteen peel, you eat it, but the peel you throw away. That is, we make the extract, and after that we add in the cosmeceutical. This is the most favorite. Aloe vera or Ghisalesa glabra. This is licorice extract. This is most of the chemicals from the plant that is to be a whitening. Yeah.
Okay, thank you, Prof. Sukanya.
Welcome.
I hope so once again, I'm sorry we have very limited time.
Yeah, I
To all
understand.
participants, I'm sorry if you have some further question, you can ask later. Yeah. So we jump to the next session. The next session is certificate for Prof. Sukanya. I would like to invite Prof. Syam to deliver the certificate to Prof. Sukanya. To organizer, please share the screen to Prof. Sukanya. Thank you. To organizer or committee, could you please share the certificate to Prof. Sukanya? So Prof. Syam will deliver the certificate. Okay, thank you, organizer. Prof. Syam, I would like to invite you to deliver the certificate to Prof. Sukanya.
Thanks a lot.
Prof. Syam.
Prof. Syam, please give the certificate to Dr. Sukanya.
Yes, Prof. Sukanya itu.
Yeah. I don't know why.
Oh,
Yeah.
thank you, Prof. Sukanya. Thank you.
Thanks a lot, but I don't know my camera why it's not true.
Okay, yeah, thank you.
Thanks a lot.
Thanks a lot.
Okay.
Thank
Hope
you,
to
Prof.
see
Syam,
you
Prof.
in
Sukanya.
Thailand.
Thank you.
Welcome.
Thank you very much. I hope one day we can meet in person, right? To discuss
Sure.
more about pharmacognosy and so on. Thank you, Prof.
Sure.
Sukanya, once again. Thank you, Prof. Syam. Okay.
Okay.
Okay, ladies and gentlemen, we will move on to the second speaker. So our second speaker today for today's webinar is Prof. Mogana Rajagopal from UCSI Malaysia. Organizer, can you please share the screen of Prof. Mogana bio? Thank you very much. So I will read Prof. Mogana bio shortly. So Prof. Morgana is currently the Dean of the Faculty of Pharmaceutical Science in the UCSI Malaysia. Prof. Morgana main expert are in pharmacology, phytochemistry, community pharmacy practice, and pharmacy laws of Malaysia. Prof. Morgana obtained her PhD from University of Nottingham, and she also holds a research grant of over 2 million ringgit Malaysia. Is also involved in national and international research collaboration. She also been awarded 12 international awards and 5 national awards, including multiple gold and silver medals in prestigious international innovation expo. So today, Prof. Morgana will deliver presentation about the application of pharmacognosy in plant-based antimicrobials and antitumors. Before we begin the presentation, if participants want to ask question, you can type the question in the chat box, and I will gladly read your question to Prof. Morgana. Prof. Morgana, you will have around 30 minutes to deliver your presentation. I'm sorry, because the time is very limited. And after that, we will have 10 minutes of Q&A session at the end of presentation. So without any further delay, ladies and gentlemen, please welcome Prof. Morgana from UCSI Malaysia. Prof. Morgana, the time is yours. Prof. Morgana, I think you are on mute.
Hi, can you hear me?
Okay, Prof, we can hear you now.
Okay, thank you. All right. Good morning, everybody. Firstly, I'd like to thank Dr. Shudin and Dr. Dia for their kind invitation. All right. So,
Okay, thank you,
I
Prof.
was yes. Dr. Dia, if you could kindly please share the slides, I would appreciate it.
Yeah, the committee, please help Prof. Morgana to share the screen.
So while the the slide is being shared, I was asked to share with all students here about pharmacognosy and basically how medicinal plants or how plants can be explored for scientific purpose. I hope today I hope how we actually look at plants.
Prof. Morgana,
Explore.
I'm sorry. Prof. Morgana, I'm sorry. I think your voice is breaking.
Can you hear me now?
Yeah, we can hear you, but your voice still breaking, Prof. I'm sorry. Hello, Prof. Morgana. Prof, I think you're still on mute. Prof. Morgana. I'm sorry, ladies and gentlemen. I think we have some technical difficulties at the moment. Ladies and gentlemen, while we waiting for Prof. Morgana connection, I will read Prof. Morgana bio again. I think I talked too fast before. So yeah, Prof. Morgana awarded in 12 international award and 5 national award. So she won the gold medal and many gold medal. Women Invention 2023 at ITEX, MTE ITEX 2022. And then she also association with British Investor and Innovator Award 2021. And also she won another gold medal in 2021 in South Korea. So I think Prof. Morgana Bio is very good. I think we are grateful to have Prof. Morgana today joining our today's webinar in about pharmacognosy. If anyone want to ask question, you can type in the chat box in English. But yeah, we are still waiting for Prof. Morgana connection to be restored. Also to all participants, I'm because due to limited time for today's webinar, I limit the question to only 5. Yeah. So I'm sorry to anyone whose question not answered before. I'm truly sorry. For the next session with Prof. Morgana, we will have maximum 5 questions due to limited time.
Oh, I'm sorry, Mr. Michael. I'm still contact Prof. Morgana.
Hi,
Hi, hi, Prof.
can you all hear me?
Yeah,
Yes.
Prof. Yeah, I can hear you, Prof. Thank you, Budia.
You can
Do you
hear
have
me?
some connection problem, Prof?
Yeah, my Wi-Fi seems to be maximum. Can you hear me? You can hear me,
Yes,
right?
yes, we can hear you.
Okay. Maybe we can proceed.
To the organizer, please share Prof. Morgana presentation. Thank you. So yeah, Prof. Morgana, the time is yours.
Okay. All right. Okay. So I'm going to tell you about how long can we apply and I'd like to share this.
Prof. Morgana, I'm sorry, we cannot hear you at the moment. Prof. Morgana, I'm sorry again to interrupt you. That we cannot hear your voice, Prof. I think the connection problem still occurs in your side. Or I think you can turn off your video to better connection. I'm sorry.
Mita to move the slide.
Organizer, please move the slide to the next slide, please. Because the slide also freeze in Prof. Mogana side. I'm sorry, ladies and gentlemen, we're still having technical difficulties with Prof. Mogana. Hello, Prof. Mogana. Hello, Prof. Mogana. I'm speaking on the Zoom.
Okay.
I'm speaking. Hello, Prof. Mogana.
I will use the audio from the mobile.
Okay. Hello, Prof. Mogana. We cannot hear you in the Zoom. Maybe you can restart on the Zoom.
Hi, can you all hear me now?
Yeah, Prof. Mogana, we can hear you now.
Okay.
Loud and clear.
Okay, so shall we proceed?
Yeah, Prof. I will
Okay, we probably can move on to the slides. Let's see, slide
this
slide
one, I
8.
think
Slide 8.
slide. This one.
Okay. Yes, thank you. Okay.
Thank you.
Apologies to everybody for the technical issue. I have no idea why, because now it seems to be clear. So anyway, so a little bit of sharing. So today I will be using cancer as an example of how we can actually utilize plants, all right, to explore various aspects of plant-based therapy. So if we look at cancer, for example, if we look at world statistics, there are about 20 million cases up to 2022 itself. And out of that, you can see about 10 million deaths already occurred. So if we look at next slide here, and in these statistics, so if we look at next slide, please. So in Asia, if we look at the 10 million deaths alone here, of course, when we look at the entire world, global population, Asia constitute nearly half of the population. And Indonesia and Malaysia definitely includes that. And you can see about 50% of the cases, incidences come from Asia. So which means that it is not surprising because of the global population, but it is a very scary prospect to know that so much of cases have arisen over so many years. And if we look at next slide, we can see that what are the current problems associated with cancer? So we know as students and those of you who are researchers, we know very well for cancer, one of the pertinent thing that is an issue is, of course, the high cost of therapy. And of course, the chemotherapy or radiation or whatever it may be, it is not a cure per se, rather a preventive measure. And of course, treatment is there, but whether it will lead to a complete removal or not is questionable, and it depends on when the cancer is actually detected. And of course, undeniably, there is high toxicity, there is a lot of side effects associated with the therapy. And one of the other thing that is a big issue is the lack of scaffold for molecules for cancer therapy. So what do we mean by lack of new molecules or new scaffold for molecules? I'll explain a little bit later. So having all this issue, of course, the entire world, when we look at over many, many years now, cancer is something that has been investigated using medicinal plants for a very, very, very long time. All right. And why? Because we are looking at, is there an alternative therapy available, something that's biocompatible, something that's cost effective, something that's eco-friendly? Of course, all of these things are what we want to look at. So if we look at this slide here on the screen, when we are looking at another measure, another alternative measure, and we look at God-given treasures that we have in this part of the world. If we look at Indonesia, Indonesia rates second in the world as the most bio or mega diverse countries in the world. And that is a very, very high figure. And we look at Malaysia, Malaysia stands at number 12. So what does it mean by mega diverse countries? The biodiversity index is so high in Indonesia, top 2 in the world, meaning that there are so much of variations in terms of natural treasures, in terms of marine sources, in terms of plants, in terms of the natural resources that are available in your particular country and in my country as well. So then if we look at the next slide here, we can see this mega diversity. If we look at Peninsular Malaysia itself, Malaysia rates as the third oldest country in the world, which is 130 million years of age, the rainforest. And needless to say, Indonesia rainforest is much, much older than that. So when you have such an old rainforest that is God-given, there are too many precious medicinal plants and natural resources over there in terms of bird species, mammal, reptilian, marine, freshwater, vascular plants, and so on and so forth. So the question is, how much of these resources have we been using? So if we look at next slide here, so when we look at the rainforest that is so diverse, again, how much have we actually explored? And over centuries, we have seen over so many years now, plants has been a very how do you say, people have explored plants for cancer. We have seen it, but the thing is, the question now that we're going to discuss today is, how successful has it been, and why have there been limitation, why have there been challenges? Look at next slide. So if you can see over so many years, you can see from 1931 up to 2013 of the data that we have now, we can see that the number of natural products that have reached clinical trial, all right, we can see are very, very few. Okay. And from this, about 38 products are for oncology or for anti-cancer. And you can see for anti-infective, there's about 26. Neurology is about 6. Immunological anti-inflammatory is about 11. And cardiovascular metabolics about 19. So the number is actually pretty small. All right. Although the investigation has been so immense. Next slide. So if you can see, even in globally, what has been done to investigate all these natural products. So WHO has this traditional medicine strategy that they have introduced since 2014 up to 2023, which is supposed to look at traditional medicine, all right, for the prevention of cancer. But the question is, how successful is this strategy? Until today is 2024, we have not seen much things happening. And one of the main reason is because there has not been, although a strategy is there, a policy is there, when there is no consolidated efforts to unify or unite everybody to bring along specific policies that can be implemented together in specific regions of the world. If this is not done, then there's nothing much we can see because everybody is working in silos or individually. So if you look at the next slide here, we know that when we look at natural product research, all right, there will be 3 phases that we look at. First is where we look at what we call the molecule phase. So the entire scientific research involving natural product, whether we use medicinal plants or whether we use certain herbs known or spices, it will what we call a molecule to bedside. So what does molecule to bedside mean? When we start looking into the plant, we have phase 1. So we'll do extraction of the plant. We'll standardize the plant. So what is standardization or fingerprinting of a plant? This is very, very important because we can be extracting many, many things from many, many plants. But if we don't standardize or we don't fingerprinting means we ensure that every time we take an extract, the same amount of chemical markers are always present. That means the quality assurance is there in each of these extract. So we actually fingerprint them. So this is very, very important to ensure quality assurance of a particular extract that we use. And then once we have this extract, standardized extract, we need to look at whether it is working for a certain disease model or whatever. So we do screening. So if, let's say, we're looking at cancer now, we screen against certain cancer cell lines. And if it is working, then what do? Then we look at pre-formulation studies. So we look at whether we want to investigate further into as the whole plant extract, or we want to fractionate the plant. That means a single extract that we take. Let's say we use chloroform or methanol or hexane extract. All right, this is what we call as whole extract. Or we want to fractionate further into different polarities, whereby we narrow down the amount of phytochemicals in each of the extract. This is called fractionation. Or we want to mix certain extracts together. So if we mix more than one extract in a formulation, we call it polyherbal formulation. Or we want to look at one compound that is extracted from the extract that we use, and this we call bio isolation or isolation of the active compounds. So there are different ways of how we can look at, and then whichever we decide, then we look at formulation development. And as pharmacists, I think most of the students here are pharmacists. One of the things that is our strength is looking at formulation development. How do we ensure that an extract can be converted into a human-friendly dosage form? Gone are the days where, we look at only tablets or capsules. Nowadays, there's many, many more innovative formulation that we're looking at. We have nano emulsion, we have solid lipid dosage form, we have microneedles, we have patches, we have hydrogels, we have many, many types. So after this is done, then we're actually looking at how does this particular formulation work. So if we look at the pharmacological or preclinical studies, which is a phase 2. So this is where cell study or animal model studies come in. And of course, when this proves to be successful, we move on to phase 3. That's the clinical study. So the clinical study here is where we look at human sample. So moving from basic in vitro, we go to in vivo, and then phase 3 is where we come to the human phase. In human phase itself, we have pilot study, we have phase 1, phase 2, and phase 3 studies. So this is generally the 3 different phases of natural product research, or what we call molecule to bedside. So we look at the next slide. So over many, many years or centuries, how have we been exploring plants, all right? Traditionally, if we look at it, we look at ethnopharmacology or ethnobotany. So what does this mean? So let's say we look at in any country that we take it, Indonesia or Malaysia. If we go to certain rural areas, villages, that would be what we call a Dukung, or some traditional healers will be there. And they use certain plants for certain reason. For example, when someone ends up with fever, they will usually take some plants and they'll make a decoction of the plants, and they say, okay, you drink this, you'll be okay. So these are different things that traditional healers use. So this knowledge that we get from these traditional healers are what we call ethnopharmacology. The knowledge that is used by these traditional healers, then what we do is we try to take this knowledge and we try to put science into it. We try to see whether scientifically what they are using, whether does this extract really reduce fever, or is it reducing some sort of is it killing some sort of virus or some sort of bacteria that's giving reduction in the fever. So we investigate scientifically. So this is the traditional approach where we have been using for many, many years now. We call it the ethnopharmacological evidence-based investigation. So then over time, I think 20, 30 years, there's also been the bottom part. We look at the modern approach where we use or introduce computational chemistry. So what is computational chemistry? If we look at next slide. So here we can see, now when we look at the traditional method that I have described just now, the traditional users use it for something, and then we now try to give some science to it, where the evidence-based, whether it really works at what they're using it for. And we look at it from the lab. All right, so we take the extract, we do the screening, and then we go into the in vitro, we go into the in vivo, and so on and so forth. And now this is not a short process. It takes a lot of time. It's quite laborious. So this computational chemistry, where technology comes in, it kind of shortens the time. So what we look at is, if let's say, once we have started the lab investigation, and we see that we isolate certain compounds from the extract, and we see that some of these compounds seem to be reducing fever in a certain mechanism. So when we start to investigate in the lab, it's a long process. We start to see whether, before we do the lab work in animals, introduce it in animal, we try to see whether it works in a software. So this is computational method. So we try to put this same molecule, all right, in the system, and then ask the system to dock to a certain protein target. That is the particular target that we want. Let's say we're looking at it in killing certain bacteria. So the bacterial protein, we start to see dock this molecule to the protein target to see whether it is
showing some affinity to it or not. So if it does, then, oh, okay, that is good. We look at how strong is giving this affinity. So that's how we predict whether it is going to work in a certain bacteria or not. And then we will simulate that in the lab, which shortens a lot of time. So then we move on from in vitro, in vivo, and clinical trial. So this is the second method that has been introduced. So if we move on to the third method, next slide. So this is different. This is also has been used for quite some time. We call bioprospecting approach, the third approach. So what does this mean? Now, this includes a lot of literature, systematic analysis of literature. So what we look at is we look at a lot of traditional compendium. So in many parts of the world, so if we take the biggest population in the world, China and India, they have their own traditional medicine compendium. All right, traditional Chinese medicine, then we have traditional Indian medicine, Ayurveda, we have traditional Malay medicine, we have Unani, the Greek and the Muslim medication. So we have a homeopathy, we have a lot of this. And most of these traditional approaches, they have their own compendium, we call it, or collection of all of the plants that they use for treatment. So based on all of this literature and those a lot of so much of articles or research articles that people have investigated, so we do a little bit of research. Let's say we're looking at a certain cancer, let's say cervical cancer. So we look at what kind of targets are there in cervical cancer, and we look at what are the plants in different kinds of treatment that has been investigated. So when we do this check in softwares, in database, in literatures, in compendium, we call it bioprospecting approach. So based on this, if we look at the right side, the cone here, from 2010 to 2017, we can see how much of literature has been published for anti-cancer phytochemicals alone. You can see the number, as you go up the cone, you can see 58.29% has been investigated for plants and cancer studies. So that is quite a lot. So based on this, we will pick up certain extracts and we will do further investigation. So this is bioprospecting approach that we can also look at. Then moving now, when we do all of these approaches, all right, next slide, please. I have a particular video, but I'm not sure whether it'll be clear. So let's move on. So the fourth approach that, next slide. We'll move on the video. I think it may not be clear. Okay, so the next approach, when we look at number 4, now this is is a very new method, quite rampantly being investigated in China and India, and a little bit so in other Western parts of the world. So what is this called? It's called network pharmacology approach. Now, what is network pharmacology approach? Now, we have seen looking at the traditional method, then we bring in the computational method, putting it into a system to see whether it's going to work before we investigate in the lab. And the third one is looking at bioprospecting literature. We look at literature and then we try to give some evidence to whatever that is in the literature to see whether it works for a certain disease model. Now, the fourth one is where we start to predict what will happen to this particular natural product or extract in the human body before we actually start to go into phase 3. So what do we mean by this? For example, we take, let's say, one compound from a plant. All right, so we have one compound, a bioactive compound from a plant that we are looking at. And before we can bring it to clinical trial, there's a lot of phase 1, 2, 3 that we need to do, right? Now, what we do is we use this compound and we do network pharmacology method using machine learning. So how do we do that? So this includes a computational approach whereby the computer guys work with pharmacologists, all right, and we also work with biochemists. So what we do is we tell the computer people, okay, we want you to create a network, all right, an algorithm, okay, whereby I want to know, I have this compound, I want to know when it goes to the human body, how does it, now I'm thinking of this compound to be effective in the heart, for example. Now I want to know if I introduce it in the human body, does it really go to the heart Or does it also go to the kidney? Does it go to the brain? Does it affect the liver, for example? Does it give any adverse effects in other organs? So I then tell the computer guys, okay, I have this protein target in the liver. I want to see whether it gives effect in the liver. And then this protein target in the kidney, does it even affect the kidney in this protein target? So by giving all this physiological and molecular targets to the computer guys, they will work an algorithm. So based on this algorithm, they will then introduce this molecule into their algorithm and to see how does it work in a human body simulatedly, how will it work? So we can see from here how the compound will be absorbed, distributed, metabolized, and excreted in the human body even before we do the lab studies. All right, so this gives a very high predictive value on how do we foresee this compound acting in the human body. So this is a very powerful tool, all right, when done correctly, and it saves us a lot of time. For example, like we know, phase 1, 2, 3 of a natural product study takes a long time. And this is one of the reason why many, if you can see over so many years, the number of product reaching clinical trials only 38. That's very, very little. But because it takes a long time to investigate. But with this kind of approach, it shortens the time by half. All right, because now we know, okay, this compound is giving effect to the liver. So then, which means that I need to reduce the side effects or to the liver, and I need it to be more targeted to the heart. So then I look at certain formulation even before I move on to phase 2 and phase 3. So which means I can preempt a problem that will already happen before by network pharmacology, all right, rather than the old-fashioned method where until human trial or animal trial, only then I discover there may be some issues or adverse effects. Okay, so this saves a lot of time. Next slide. So then another issue that can be very much queried is that the sustainability of this. Now we have Indonesia, topmost mega diverse, highest biodiversity in the world, Malaysia number 12. Now with all of this, we cannot be going and destroying the particular plants all the time, right? Then we have very high deforestation happening. So then to avoid this, what some people do is, if let's say we are looking at one molecule, isolated molecule, instead of us cutting down all the trees to get as much as yield as possible, if we already have a certain pathway of how we are isolating or getting the particular compound for from the extract, for example, we look at system biology or synthetic biology or metabolic engineering. So what does this mean? That means, can we produce this molecule in the lab rather than destroying all the plant? Because now we already have a scaffold. So this goes back to the beginning of my lecture that I talked about scaffold. So the whole problem with drug therapy right now, even if you look at cancer, for example, is lack of scaffold. Scaffold means what? We look at building, when we build an infrastructure, we call it scaffolding. You build the scaffolding or the framework. So similarly, in drug therapy, lack of scaffold means that we lack novel or new kind of molecule framework for certain therapy or certain treatment. We only have the same molecule type that we work at, and synthetic chemists use it, change the functional group to see, will it work? If I change this functional group, will it work in this way? If I change this functional group, will it work in this way? And we go on changing that. But we lack new novel molecules. So by doing the isolation method, if we come up with certain novel molecules, we cannot be deforestating the whole area. Rather, we then go to the synthetic chemists. Can you please produce this in the lab? So that is what we mean. We need to look at new scaffold molecules. So then looking at the next slide. So if we see an example of how the network pharmacology has been very useful and powerful. So example, this cancer HSP example that I like to quote here. Using this system here that they developed, this algorithm, they were able to quantify or to consolidate, 2,439 anti-cancer herbal medicine with over 3,000 anti-cancer ingredient, all by using the algorithm. All right. And then they're able to identify 9 ADME parameters by looking at the absorption, metabolism, excretion, distribution, and so on and so forth. So this gives a very good background data before the pharmacologists look at the actual lab work. All right, because they already know they predict how these molecules will already work in the body and with the different targets in the body. All right, next slide. So just this, like, I like to quote this since I don't have much time. So for example, we have seen how medicinal plants or natural product can be explored, the various areas. So it opens up your paradigm of how you can look at plants in investigating different modules, how you can explore them. All right. Now, there are three main ways of how we can look at plants per se. All right. One way is the bioactive compound. That means you have an extract and you want to look at it as a cancer. Let's say you want to see whether it's effective against cervical cancer cell line. So example, CH cell line, and you run through the CH cell line. And then from there, you have bioassay guided fractionation, and you come up with one single molecule. So this is what we call bioactive compound. All right. Number two of how we can utilize natural product research is that as bioenhancers. So what does bioenhancer mean? For example, let's say I have this particular molecule, all right, and I see that it is very effective against killing CH cell line or the cervical cancer cell line. And then what I do is, if I combine this molecule with another known therapy, known drug therapy for a cancer, and to see if I'm giving it together, will it will it be effective in killing the cancer cells more effective than either alone? So this is what we call as bioenhancers. So bioenhancers, what do they mean? They work synergistically. They give better effect when used with a known therapy, or they could also be used to increase the bioavailability of an existing therapy. All right. For example, if I'm mixing compound A from a plant with a known drug A, all right, and this compound that I'm isolating and I'm combining with drug A increases the bioavailability of drug A to the target area, which means it is effective, much better than used alone. So bioenhancers. That is number two. Number three of how we can use it as polyherbal formulation, PHF. What does this mean? Combination of more than one herb is called polyherbal formulation. This is very popular in Ayurveda and traditional Chinese medicine. And of course, in all Indonesia, we have jamu. So jamu is a mixture of certain herbs together. And this is an example of polyherbal formulation. Now, why do we mix all this? Is it just like random mix? No, there is a science behind mixing these herbs. And this is what we call polyherbal formulation, whereby when we mix it together, the reason is we want it to work together better, synergistic effect. Because when we look at therapy, when we take one herb, let's say it gives you an anti-cancer effect. But when it gives you the anti-cancer effect, maybe it gives you side effects together. And then you put in a second herb to increase the bioavailability of herb A to that target area. But then what about the side effects? So you put in herb 3 to reduce the side effect caused by this herb 1, for example. So in the end, you have a package formulation, polyherbal, whereby not only you are reducing the killing of the cancer cell, at the same time, you are reducing the side effects that may arise because of this killing. All right. So, which means the complete package. So, this is a polyherbal formulation, the benefit of using polyherbal formulation. So, these are the three ways that we can look at natural products. And of course, being pharmacists, most students here, you're pharmacists yourself or student pharmacists, we need to look at gone are the days where we only look at capsule or tablet. All right. Now we have so many formulation that we can explore. An example on the screen, you can see polymeric nanoparticle, SLN, liposomes, microsphere, nanoemulsion, mucoadhesive nanoparticle, nanocrystal. It's just endless. Sky is the limit. There are so many investigations that can be done as pharmacists, all right, in utilizing these herbal products in three ways: bioactive compounds, bioenhancers, or as polyherbal themselves. Next slide. So when we look at next slide here, all right. So plant, like I said, when we mix more than one plant together, it is as bioenhancers synergism. And then what do they do? Increasing bioavailability, permeability boosters to increase the drug concentration of a major component, for example, so on and so forth. Next slide. So just to share very quickly, I'd like to share this example of this particular polyherbal formulation that we have formulated as solid lipid nanoparticle. So when we formulate as solid lipid nanoparticle, we see that the entrapment efficiency or the drug entrapment is very high, up to 92%, which is very high. So, and what this does is there's very high bioavailability of that product, and it's biodegradable without formation of any toxic degradation products. So these are the benefits of different kinds of formulation that we can look at when we are using polyherbal formulation. Next slide. So this is another example of when we look at polyherbal formulation in colorectal cancer. All right, how it actually inhibits HCT colorectal cancer cell compared to 5-FU or the known therapy. Next slide. So here is another example of TC16. This is a polyherbal that when we mix with a polymer in an aqueous oil phase, and we have a nano emulsion. So of course the nano emulsion can be freeze dried or spray dried, and then you have the powder that you can then look at encapsulating it. And the benefit of this formulation is you can see small droplet size, large surface area, low viscosity, protects the active compound. So in this way, the bioavailability of that product will be higher. Next slide. So then when we take this nano emulsion and we look at fibroid cells or EL3 cells, uterine fibroid, women have this as a major problem, but there's no cure until today. So when we can see that it actually inhibits the EL3 cells. Next slide. Next slide. So then I'd also like to open up the paradigm to another factor here. Now, we all know that animal study or animal model is a very important step or phase 2 study that we look at before we go to human study. Now, and what happens in this animal study is we kill a lot of animals. All right. So each time as pharmacologists, minimum we'll kill 120 or more rats or mice. And this is very rampant. It's quite cruel in a certain way, but we can't help it because it's for scientific purpose. But we can see many regions in the world have start to look at, can we look at a method where we don't kill so many animals? So this is where pharmacoscintigraphy imaging comes useful. So what is this? Now, in pharmacy, one of the field is radiopharmacy, where we look at isotope labeling for diagnostic purpose. For example, when we look at rheumatoid arthritis, how do we diagnose rheumatoid arthritis? We usually radioisotope tagging. We tag it and then we introduce as an agent into the body for diagnostic purpose to see where is lodged, and then we can it will assist the medical specialist to diagnose a certain disease. So same thing, we also look at using the same technique for science. So example here, technetium-99, we radio label against an herbal product, for example, or herbal extract. So when we radio label it and we introduce into the animal, usually we use a rabbit. So when we use rabbit, we can see when we radio label the radioisotope with the herbal product that goes into the animal, we can look at the imaging and we can see where does this product go. If I'm claiming that the product doesn't cross blood-brain barrier, doesn't have any effect on the brain, but it is very targeted to the heart, for example, does it really go to the heart, or does it get deposited in the brain? Does it get deposited in the kidney? And we can look at the imaging, the gamma imaging. So this will help us a lot, not only to tell us whether the product is working in animal, at the same time, where is it deposited in different body parts of the animal. So this, of course, shortens a lot of time And then we can go back and look at if it really goes, instead of being targeted to the heart, it now being distributed to the kidney. Then we say, okay, we have a problem now. We need to see how to solve this problem because it is distributed in the kidney right now and we don't want that. So this is an example of utilizing radio pharmacoscintigraphy imaging to shorten the time in natural product research. So with these examples, when we look at next slide, the future prospect that we want to look at is utilizing a variety of all these approaches and of course technology. When technology comes in, we cannot say I'm not going to use technology. We must learn how to embrace technology to shorten time and looking at all the God-given treasures that we have in nature, because there are so many diseases out there, but there is also a lot of unexplored plants out there that we need to look at. And how do we look at sustainably? We use technology. So extensive research work needs to be carried out in a variety of ways. We should not only be looking at bioactive molecule, we should investigate as bioenhancers, we need to investigate as polyherbal, broaden the horizon, broaden the paradigm of research, not fixed to one particular pathway per se. So when we look at, so that is roughly what I hope that I have given a little bit of overview of how we can use pharmacognosy in terms of investigating a specific disease model. So next slide. So I thank everyone today, and this is my team here. This is what we call our Women's Health Research Team, because main part of our focus is on women's health. So we look at cyst, fibroids, cervical cancers, primary dysmenorrhea, and so on and so forth. And what we look at are actually bioenhancers, bioactive molecules, as well as polyherbal formulation. And some of these products has been formulated, and part of the award that has been granted to the team comes from these polyherbal formulations, which are also some of them in the process of commercialization at the same time. So thank you very much, everyone. Appreciate your time and appreciate for inviting me. And again, apologies for the technical error in the beginning. Thank you.
Thank you, Prof. Mogana. Thank you for your very interesting presentation. So if I may summarize a little bit about your presentation today. Your presentation today talk about the newer approach to herbal research such as network pharmacology, or on the last slide, you talk about Techhum-99 to radio label the herbal. So yeah, it's very interesting that this method can make the product faster in the market, of course, and can be used by patient for treating of some disease such as cancer. So yeah, Prof, I would like to read some question. I think we already have some question in the chat box. Okay, the first question will be from Salwa. So, Prof. Mogana, are there any risk or side effect to consider in anti-cancer treatment, such as drug interaction or tolerance? What are the advantages of using medicinal plant as anti-cancer compared to conventional therapy? Conventional therapy, I think you mean drug, right? Thank you, Prof. Mogana. Please answer the question.
Okay, thank you. I think the question is asking about the effects of conventional therapy. I think I've shared in the beginning of the presentation. I think it's undeniable. We are all in the healthcare, and we know very well the adverse effects of all these therapies. Because when we look at cytotoxic compounds, not only the problem is always with the selectivity, because when it is supposed to be killing the cancer cell, they will also be killing the normal cells. And because they do that, the side effects of that particular therapy not only kills the cancer cell, but because it kills the normal cells, all right, the side effects then precedes the killing effect here. So, and that is a common thing that we see, and that's where you have all the side effects of the vomiting, losing of hair, and the degeneration of bones, the lethargic effects, and so on and so forth that we see. So this is a known factor. So now, why, when we look at natural products, what we are aiming for here Can we get higher selectivity with natural product? Can we look at combining a few of these extracts together as in polyherbal formulation, whereby not only it kills the cancer cell, all right, it is targeted. At the same time, if there are certain side effects arising from it, also killing a normal cell, we have another extract here to reduce the side effects. So when we are giving it to a patient, the patient does not experience as much side effects as would be seen in a conventional therapy. Because at the end of the day, especially for cancer patient, for example, is the feasibility, the convenience, lifestyle, how much are we going to ensure that it doesn't affect negatively in their lifestyle? Because quality of life is very important, and this is very low in cancer patient, because in any therapy the quality of life is drastically reduced. They go into an uncomfortable situation, a lot of pain, a lot of effects that they have to put up with the side effects of the conventional therapy. So we want to look at a polyherbal, for example, that when given, not only it is able to increase the killing effect of the cancer cells selectively, also does not affect their lifestyle too much. That means less side effects, because we have the other extract here to counteract any side effect that may occur. So this is what we are aiming for. Hope that answers the question.
Okay, thank you, Prof. Let me move on to the next question from Adi Dwi Margono. The question is, how does the extraction process and standardization in phase 1 contribute to the effectiveness of clinical studies in phase 3 of natural product research?
Okay, thank you. Okay, thank you for the question. I think standardization and fingerprinting is absolutely essential, because one of the major queries that Western people will have against Asian medicine, traditional medicine, is how do you know that the particular extract that you're using today, it will be the same as the extract that you're using tomorrow? When they say same, meaning that the content or ingredients or the phytochemicals present in that extract will be consistent. This is one question that Western practitioners always question in traditional practice. So this is why standardization and fingerprinting is absolutely pertinent. Because when we fingerprint, we ensure that every single extract that we use anytime, all right, is reproducible and has the same composition or ratio of phytochemicals that we are looking at. So when we are able to ensure that the quality is always there in each extract, when it reaches up to clinical trial, when it's given in the human, we can then predict that the effect is very consistent, because the extracts that we're giving to the humans are consistent in the phytochemical composition. Otherwise, it may result in a way where it may work certain times, it may not work at another time, because the composition of phytochemicals in that extract is not the same. So if the phytochemical is actually giving an activity, a therapeutic activity, but it is not standardized, so it may give you effect sometime, it may not give, because then the ratio is less than what it's supposed to be, may not give you the effect. So it is very important that reproducibility is there, and that is why fingerprinting and standardization of extracts very important to ensure that when the product reaches human trials, the activity is always consistent. That is very important.
Okay, thank you, Prof. I hope that answered the question from the participant, from Adi Dwi Margono, right? So I would like to next move on to the next question from Megawati. Could you describe the role of each stage in the future for the development of computational approach and bioactive compound, and how does they collectively contribute to improve the efficacy and safety of clinical trial? Thank you.
Could you describe the role each stage envisaged in the development of computational approach to bioactive compound, and how they collectively contribute to improving? Not really sure what is being asked here. Basically, what I have mentioned just now is how each stage when we are starting the phase 1, stage 1, stage 2, stage 3, right? So if we are asking where is the role of computational approach is always towards what we call the um, predicting how a compound works in a simulated area before we look at it in the wet lab. So when we look at it, if we already have an isolated compound, before moving into the wet lab, we will do the computational to predict how it's going to work. All right. And we can also introduce network pharmacology. And then we duplicate or mimic that into the wet lab to see whether there is a consistent outcome that we are going to look at. And of course, when we have done all this, at the end of the day, this is an algorithm. Computational chemistry or network pharmacology is an algorithm. It enables us to predict or forecast, all right, or shorten time. But it is not limited to that as well. That means it may not have 100% accuracy, of course, all right, but it enables us to predict or forecast what kind of side effects that we may see in a particular treatment, what kind of therapeutic effect that we predict that can be seen in a particular treatment. It is a predictive measure. It shortens time, all right? But it's, of course, it is not 100% accurate as all machine or algorithm or AI technology that we are looking at. Hope that answers the question.
Okay, thank you, Prof. Morgana. I think we will jump to the last question of this Q&A session from Alfi Kusuma. The question is, how does bioprospecting approach relate to the development of new drugs? Thank you.
Okay, thank you. Okay, so let's say, let's use an example. I'm bored with maybe not a cancer example. Let's go into a bacterial example, for example. Let's look at MDR, multi-drug resistant bacteria. So we know that MDR, you have Klebsiella, we have Acinetobacter baumannii, we have MRSA, whichever, which is a lot of these bacteria are now becoming very resistant to therapy. So let's say we are looking at an MRSA factor, and we are looking at how do we find new antibiotic that is plant-based, all right, rather than, because we don't have this. It has been more than 20 years where a new antibiotic has come into the market, and the reason why, because we don't have any new scaffold of molecules, all right, and the reason being the traditional approach needs to be more explored, all right, and we don't have new. So synthetic chemists are working with the same molecule, looking at changing functional groups to develop new kind of drugs, but still we don't have new antibiotics in the market. And resistance is increasing so much that by 2050, we are looking at over 10 million deaths because of bacterial infections. So this is very grave. All right. So when we look at bioprospecting, so bioprospecting method, if we look at all of the studies that are being done, we look at compendiums. When we do a very systematic research on this literature, and we look at shortlisting certain candidates, certain plant that may have effect on a certain MRSA, for example. That means they have preliminary work done. Traditionally, they have used for certain kind of MRSA-related diseases. And we then shortlist all of this. And then it gives an idea, rather than starting from nowhere, we have no idea where to start to target this MDR bacteria. But when we do bioprospecting from different compendium literature, published articles, we shortlist the amount to certain plants, for example. And then we can now focus on this particular plants, looking at approach against these MDR bacteria. So this is how bioprospecting helps us, because it narrows down the huge database of plants that we have been investigated. It narrows down this to exactly what we are looking for, so that we can focus on this rather than we are blind and we do not know where to start from, or we start from something and we ended up having negative results. So we look at all of this published articles or compendium, and then we narrow down, and then we focus on this particular bacteria that we're looking at. Thank you.
Thank you, Prof. Morgana. I'm afraid our time is very limited, Prof. Morgana. Our time is running out, so sadly we must close today's Q&A session. For all participants, I would like to say thank you for your participation during this webinar. I'm sorry, I apologize that I cannot discuss your question due to limited time. So next we will have certificate appreciation to Prof. Morgana. I would like to invite Dr. Dia to deliver the certificate to Prof. Morgana. To the organizer,
Okay.
could you please share the certificate?
Prof. Morgana, on behalf of Fakultas Farmasi Universitas Pancasila, I would like to thank to you for the great and insightful topic ya. That was a very comprehensive and interactive presentation. Okay, I know we appreciate your lecture with a speaker certificate. Thank you.
Thank you so much. Thank you. Appreciate it.
Okay. God bless you, Prof.
Thank you so much, Dr. Dia. Thank you so much. Have a good day.
Yeah, thank you, Prof.
Okay,
Morgana,
thank
Dr.
you.
Dia. I hope someday we can meet in person, Prof. Morgana. Take
Definitely.
care.
Welcome you all to our place.
Okay, Prof. Morgana, take care. See you.
Bye-bye.
Okay,
Bye.
ladies and gentlemen, I think we have reached the end of today's webinar. I would like to say thank you for the opportunity to be your moderator today. I apologize if during this webinar I made some mistakes. Next, I will hand the webinar back to the MC. Thank you, everyone.
Okay, thank you so much, Mr. Michael, for your time. What an amazing discussion and insightful, of course. Thank you for all speakers, moderator, and participants that partook in this Q&A session. Next is a handout of certificate of appreciation to our amazing moderator who lead our session properly. And we invite Dr. Dia Ika to hand out the certificate.
Okay, I would like to thank to the moderator, Mr. Apotheker Michael. You're doing great to manage this lecture this morning. Yeah. Thank you for spending your weekend with us, and this is appreciate from us as your contribution as a moderator. Thank you very much.
Thank you, Dr. Dia. Thank you.
All right, thank you so much.
Amalia, thank you.
Okay, thank you so much, Dr. Dia, and also Apotheker Michael for your time. And by that marks the end of our lecture today. Representing the committee, I would like to thank you to all speakers and participants that have joined this lecture truly. We would like to apologize if there are any mistakes and along the way. Once again, thank you so much for your attention. I'm Amalia Zahra as the Master of Ceremony, signing out. Wassalamualaikum warahmatullahi wabarakatuh. Shalom, Om Santi Santi Om, Namo Buddhaya, Salam Kebajikan, Salam Pancasila.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih. Thank you.
Thank you, everyone. Goodbye. Have a good weekend.